of experiment. Our objectives include to identify the need to use design of experiment in product and process research and development, to illustrate the design of experiment methodology, to summarize the goals and uses of design of experiment, and to explain the steps in design of experiment. Design of experiment is a body of knowledge based upon statistical and other scientific disciplines for efficient and effective planning of experiments, for making sound inferences from experimental data. It's used to evaluate which process inputs have a significant impact on the process output and what the target level of those inputs should be to achieve a desired result or output. Design of experiments defines the population to be studied, randomization process, administration of treatments, sample size requirement, method of statistical analysis. We will look at some basic principles of design of experiment. Randomization is a process of assigning treatments to the experimental unit. And by experimental unit, we mean the smallest division of the experimental material. And when we talk of treatment, it's a specific combination of factor levels whose effect is to be compared with other treatments. And the purpose of randomization is to remove bias and other sources of extraneous variables which are not controllable. Randomization accompanied by replication forms the basis of any valid statistical test, and therefore for any experiment, randomization and replication is very important. There are many ways by which we can randomize the experiment. We could use a computer program, we could use random number table, which we could use Excel to generate, or we could use some form of physical mechanism. For instance, if we had 6 runs of an experiment, we could take 6 slips and put in a box and then choose from the box, and whatever we choose for that particular day becomes the experiment to run, as opposed to running the experiment in sequential order. This will help us eliminate human influence on the experiment. Replication is independent repetitions of an experiment under identical experimental conditions. An individual repetition is usually called a replicate. And the purpose is to secure more accurate estimate of the experimental error, a term which represents the differences that would be observed if the same treatments were applied several times to the same experimental unit. To decrease the experimental error and thereby to increase precision, which is a measure of the variability of the experimental error. And it's also to obtain more precise estimate of the mean effect of a treatment. All external sources of variations are not usually removed through randomization and replication, and therefore there is the need for balancing, blocking, and grouping of the experiment. So when we talk of local control, we're actually referring to the way we could balance the experiments, block, where we could use block or group the experimental units. Balancing is simply the treatment should be assigned to the experimental unit in such a way that the result is a balanced arrangement of the treatments. In blocking, experimental units should be collected together to form relatively homogeneous group. A block is also a replicate. Blocking removes the effects of a few of the most important nuisance variables. Randomization reduces the contaminating effect of the remaining nuisance variables. However, not all is removed, and therefore the general rule is block what you can, randomize what you cannot. And the purpose in all this is to decrease the experimental error. In all experiments, generally, we want to find a relationship between the input variable and the output variable in order to be able to understand the process which we are seeking to understand. So there's always a process we don't know fully how it operates, and for us to be able to understand it, we will usually have some independent variables or the input factors, the control factors, which we are able to manipulate at will. And once we do that, we tend to measure something. And that we measure we call the output, or the response variable, or the dependent variable. Now, we don't have the leeway to control this. For example, if you had measured the pH of a substance, whatever the pH meter tells you becomes the reading. You have no right to manipulate what you measured. So the independent variables are not usually under the control of the experimenter. And we do that to be able to link what we put in to what we measured in order to understand this process. However, there are some other variables which we are not interested in, and yet they have a way of influencing the experiment. And therefore, for any experiment, before we set out, we need to identify these variables. The ones we can manipulate, the independent variables, the ones we want to measure, and the ones we cannot control, we call them extraneous variables, and yet they influence the experiment. We need to understand that so that we will know how to handle them in order not to influence the experiment adversely. Experimental data are used usually in order to understand the process to derive an empirical model that links both the output and the input. And these empirical models can contain first and second order terms. And the two popular ones we will be considering in this course is the linear and the quadratic form. The linear form generally is that relationship that follows a straight line between the input variable and the output variable. And the equation of a straight line, we are told, is y equal to mx plus c. The y is the dependent variable, the one we measure, and our x is usually the independent variable, the one we manipulate. So in the case of equation of a straight line, we only had one independent variable of interest. However, there are cases where we will want to study more than one independent variable. In that case, the equation of a straight line would translate to this form, in which case we have the y, which is our dependent variable which we measured, and we have two independent variables, x1 and x2. The beta 0 and the beta 1 and the beta 2 and the beta 1, 2 are actually the effect of each of these variables on the dependent variable. Now, in some experiments, the relationship may not be linear. It may be a quadratic form, and therefore, in that case, we have to include these quadratic terms into the linear form in order to understand the process in question. We would look at this more when we get to analyzing these experiments. The uses of experimental design, there are many uses, one of which is to choose between alternatives. In that case, we design a comparative experiment. If our objective is to select the key factors affecting a response, we need to design a screening experiment. At some other points, the goal is to optimize, to find a target, to find the best, the minimum, the maximum response, to reduce variability. To make a process robust, to seek a multiple goal, in that case, we need to design a response surface experiment. Some other time, we want to constrain a proportion. We want to mix ingredients in a particular proportion, constraining it to say 100% or 1. In that case, we need to design a mixture experiment. At some other point, we just want to understand the empirical equation relating the input and the output. Of course, we do regression modeling, and usually regression modeling is part and parcel of response surface methodology, and we will discuss this as we get to later part of the course. Steps in achieving a desired design. The first is to identify the objective. Once we set the objective and then we select the experimental unit, it will then help us to understand the type of experiment we should design, and therefore we select an experimental design. So this experimental design depends on the type of objective we set out to achieve. And then we execute the design. We check that the data are consistent with experimental assumptions. We analyze and interpret the results, and we use or we present the result, or sometimes the result may lead us to running more experiments. So the first objective, as we said, is comparative objective. Here, the primary goal of the experiment is to make a conclusion about one important factor. So if you have one or several factors under investigation, you just want to know, make a conclusion about one of them. The question of interest is whether or not the factor is significant. In that case, we need to design a comparative experiment. An example is to compare the results from two different types of flour in cake making. If the flour from different vendors did not differ significantly, we could select the lowest cost vendor. If flour were different, then we could select the best flour. The experiment should allow us to make an informed decision that evaluates both quality and, of course, cost. If our objective is to identify the significant input factors affecting a response or an output, to select or screen out the few important main effects from many less important ones. If that's our objective, then we need a screening experiment. And in a screening experiment, we also call it main effect design. An example is what are the significant factors beyond flour, eggs, sugar, and baking in cake making. So we could screen out for these ingredients in order to identify those other factors that will have an effect on the cake making. If our objective is to find an improved setting, like minimizing or maximizing a response, for example, how can the cake be made as moist as possible without disintegrating? Or we want to achieve an optimal process output, or want to make a product or process more robust against external and non-controllable influences, or balance trade-offs when there are multiple critical to quality characteristics. In that case, we need a response surface design. In that experiment, it will enable us to estimate the interaction and even quadratic effect. If that is the goal, then response surface methodology is required. At other times, we want to know the best proportions of the factors so as to maximize or minimize a response. Here we are talking about proportions. We're optimizing responses when factors are proportions of a mixture. In that case, we need a mixture design. Regression objective is when we want to model a response as a mathematical function of a few continuous factors, and we desire a good model parameter estimate. In that case, we need to design a regression experiment. So having set the objectives, we then go into select the experimental unit. An experimental or sampling unit is the person or object that will be studied by the researcher, that is the smallest unit of analysis in the experiment from which data will be collected. Then we select the process variables. Specifically, variables represent persons or objects that can be manipulated, controlled, or merely measured for the sake of research. Now, within these broad category of variables, we have different types. So, process variables, we could have the input variable, which is the independent variable or the factors, and we have the output variables, the ones we measure, which is the responses and the dependence. The independent variables that is manipulated by the experimenter will have several different values, and these different values are called the levels of the independent variable. So in designing an experiment, we need to define our independent variables and how many levels of each of the variables are we interested in. And there are other variables that do not vary, and we call them constants. Again, we need to also understand and identify those variables we may have to keep constant in the experimental exercise. We have the nuisance factors or the nuisance variables. These are independent variables that are not of interest but could cause variability in the experiment. They are independent variables not being tested and held constant during the experiment to prevent their influence on the dependent variable. The control variable answers the question, what do I keep the same? Types of nuisance factors and cures. There are known and controllable factors. Here we use blocking to systematically eliminate the effect. So we know them and we can control them. They will be easy to eliminate. There are some others, known but uncontrollable. These independent variables known to exist, but conditions prevent them from being manipulated. If we can measure them, we go on and measure them, and then we deal with them as we go into the analysis using analysis of covariance. The unknown and the uncontrollable ones, we don't know about them. We may not even control them. We could randomize to deal with them. We have examples of nuisance factors like specific operator, time of the day, the experiment was run, panelists, and the room temperature. All experiments indeed have nuisance factor. Therefore, decide which nuisance factors are important enough to keep track of or control if possible during the experiment. The experimental error is due to the influential effects of uncontrollable variables. Experimental design should eliminate or control these types of variables as much as possible in order to increase confidence in the final results. The treatment structure, we need to select the independent variables which we need to manipulate are expected to show the effects of interest on the dependent variables. So therefore, the levels of these factors of interest should be defined. The levels can represent the entire range of possibility, or is a random subset. In that case, we have two types of levels: the fixed effect treatment factors or the random effect treatment factors. The fixed effect treatment factors are levels of interest that are included, all levels of interest are included in the study. Only small number of inherent levels, for example, only relevant levels of gender are male and female. Only certain values of the levels are of interest, even though other levels might exist, especially where regulation or legislation will not allow the inclusion of other levels. Therefore, the experimenter selects what levels to experiment on. In that case, such variables are referred to as fixed effect treatment factors. We have the random effect treatment factors. Here, levels are selected by a random process from a much larger population of equally usable levels. Then we go on to select the experimental design. And that's the purpose of this course. We've just gone through the introduction to experimental design, and that's our lesson 1. And where we have one factor of interest will take us to designing a simple comparative experiment, and that will form lesson 2. Where we have more than one factor and all the factors are 2 levels, then we will have a screening experiment, and that will form part of our lesson 3 and 4, where we look at factorial experiment and fractional factorial experiment. And when all the factors are not at 2 levels, we look at if the factors are quantitative. If they are not quantitative, all of them are not quantitative, then we look at lesson 5, where we deal with the general factorial. And if all the factors are quantitative, we look at if the effect of factors are for a process or for a mixture. If it's in a process, we will look at lesson 6, where we discuss response surface methodology. And if it's in a mixture, we will look at lesson 7, where we discuss the mixture design. And so if you're a researcher, you would want to get the skill of designing an experiment. And once you have this skill, I promise you, experiments will not be a problem for you. And in particular, when we get to the analysis of these data we will collect, if you're able to get the skill of data analysis, numbers will not scare you. And so I'm hoping you will come with me as we explore this course. Thank you very much for your attention.